the conducting system and its role in coordinating the cells of the heart to beat as a unit. And so the conducting system is conducting an impulse, a stimulus. So if we look at the anatomical picture of the heart here on the lower left down here, right, we can see that the conducting system is comprised of the SA node here in the right atrium. The atrial cells are all connected by gap junctions. That signal starting at the SA node will be conducted to the atrioventricular node found at the bottom of the right atrium. And then it's going to pass along this AV bundle right here, right, into the ventricles. And in a process of doing so, it's going to cross over what's called the fibrous skeleton. And that's a really important structure because it's an electrical insulator between the atria and the ventricles. And I'm going to come back to that in just a minute. So the AV bundle is the only pathway for a stimulus to get from the atrial cells to the ventricular cells, and again, into the septum initially. And then the AV bundle is going to split into left and right bundle branches, and then once it reaches the apex of the heart down here, right, you can see that they sort of start to turn around and come back up the conducting system fibers, and they're called Purkinje fibers. And those are sort of within the walls, but also largely laid sort of on the interior surface of the heart muscle. Like they're very superficial, not on the surface, but at the surface of that cardiac muscle. And so the stimulus is actually going to travel from the inside of the heart toward the outside of the heart, across the wall of the heart. In other words, it's going to travel in this direction, as well as, right, so at a given spot, it's going to travel through the wall, but remember, it's coming down the septum, the stimulus, and then it's going to come up the walls, right, via these Purkinje fibers. But remember, as it comes up the walls, it's also going to travel through the wall from the endocardium, the inside, to the outside. That's relatively important for some other, for some things we won't talk about right now. Now, I wanted to start with this anatomical description and layout of the conducting system so that we could move over here on the right to a much more schematic version. And so we can sort of like forget all the fine details of the heart structure and focus just on the conducting system and sort of where the signal is going. So remember, we're starting up here. Our signal starts at the SA node. And the only reason it starts at the SA node rather than any other conducting system cell, which also spontaneously depolarizes, is because these cells are the fastest cells to spontaneously depolarize. In other words, their action potentials look like this, right? When we get this pre-potential phase right here that has a relatively steep slope. And what that means is each action potential is relatively close to the next one, because it's not a prolonged period of pre-potential. Okay, and again, the nodal cell electrophys presentation talks more about that. So because the SA node fires first, it generates that signal, and it's going to travel then cell to cell. So right here and everywhere else, I have this sort of hash mark indicators. We have gap junctions, right? These are the gap junctions of the intercalated discs. I think I have them labeled down below. Right? Gap junctions at the intercalated disc that are going to convey the depolarization from one cell to the next. And so our stimulus is actually going to travel from the SA node, right, outward through the atrial syncytia, right? And again, a syncytium is a group of cells, a functional syncytium specifically, is a group of cells that are linked together by gap junctions. And what that means is that they tend to function as a unit. The desmosomes are also important because they hold the muscle cells together, but this is the system, the conducting system, delivers the stimulus to the contractile cells, right? Remember, these cells, the conducting system cells, aren't really contractile. They're muscle cells, but they are adapted specifically to carry this stimulus from cell to cell to cell quite rapidly. Now, notice I have out here, we're going to go from the conducting system cells to the cardiomyocytes, right? And so it's going to go from conducting system cells, and then in the later dominoes, if you will, most of the dominoes actually are cardiomyocytes, which are also linked by gap junctions, right? But so we've got these preferential conducting system pathways that lead to the cardiomyocytes to generate their stimulation and contraction, right? And so we're also going to travel down to the bottom of the atria, specifically the right atrium, as we mentioned over in the other figure, and we're going to run into the AV node. And the AV node also conducts this stimulus, and again, it's going to do so through, very importantly, it's going to do so through this fibrous skeleton, right? This is I want to go over here, right? It's going to do so through this fibrous skeleton of connective tissue, which is an insulator, right? It does not conduct action potential. There are no gap junctions that pass through it except right here, right? So we've got this one gap in the fibrous skeleton below the AV node where the AV bundle will pass through that fibrous skeleton. That's really important, right? For a couple of reasons. One is that the AV nodes delays the signal, right? So the atria, in response to the SA node action potential, the atria will depolarize and begin to contract. But because our stimulus is delayed at the AV node down here, right? We have a delay here generated. It's going to separate in time the atrial contraction from the ventricular contraction, right? Hugely important. Remember, the atria, their job is to top off the ventricles in terms of their filling. You can't fill them, the ventricles that is, you can't fill the ventricles while they're contracting, right? They have to be relaxed. And so the atria have to contract before the ventricles start to contract, right? So this delay helps with that. It separates the atrial and ventricular contractions in time. That's key, right? So then, it's a very short delay. Remember, you've got a full cardiac cycle per second at rest, about 1 beat per second, 60 per minute. And so it's a short delay, but it's enough to separate those two events. Once we go down, now we would be in the septum here, right? And we're off to the races again. We're sending our signal down to the apex of the heart and then back up through those Purkinje fibers and then off to the cardiomyocytes of the ventricles, right? So we essentially have two syncytia of the heart, right? We've got an atrial syncytia and we have a ventricular syncytia, right? Right? And they are separate but connected, again, by this one connection across the fibrous skeleton via the AV bundle, right? So we get a delay, and then we get passage through that. But it's important that this fibrous skeleton does not allow the signal to pass anywhere else because, number one, it would pass through much more quickly, right? Because we would not have that AV node delay. And secondarily, it would pass through and start depolarizing the heart in an abnormal pattern, right? We want it to come down the septum and then up the walls. We don't want it to sort of come down, somewhere else, right? You don't want it to cross over here. That would be bad, right? Because then we would, it would potentially start to depolarize over in this region and first, and that's not what we want. Remember, electrical is the stimulus for the mechanical. Whatever depolarizes first, contracts first. And of course, we want it to contract in a particular sequence so that we can move the blood from the atria into the ventricles, and then from the ventricles out into the pulmonary and systemic circuits. Okay, now one other thing about this conducting system before we move on is that it is innervated. right? This is not a system that is acting in isolation. Make some room here. Right? There is input from the nervous system in the form of neurons that come down and synapse on these structures. Right? I'm actually going to have one come down. It's not going through that cell. I'm just sort of crossing over here. Right? And so this is my neuronal shorthand here. Right? There's a soma. There's an axon, and then this V at the end is the synapse. Right? And so we have two different groups of cells that are going to innervate these structures. Now, the blue one I'm going to put coming down here as well. Right? It's going to innervate the AV node. And honestly, I should have had that one come down even farther. Right? This guy's going to come down. You know, I'm just showing branches of it. It doesn't have to be a single neuron doing this, but we get axons that come down and innervate the ventricular cells as well. All right, so what are these two neurons that I drew? Again, representative neurons that I drew. Well, they are the sympathetic and the parasympathetic innervation of the heart. Right? The sympathetic, I'm going to have be this sort of purple color, and it's innervating the SA node, the AV node, and the ventricles. Notice the parasympathetic neuron I drew here, representative neuron I drew here, innervates the SA node, the AV node, but it doesn't really send any axon branches down or separate axons into the ventricles. And that's true. This parasympathetic doesn't really alter the activity of the ventricular muscle directly. So what that means is that the sympathetic and parasympathetic, right, are both innervating the SA node, which means they both play a role in determining heart rate, because our heart rate is determined at one location, the SA node. Innervating AV node can adjust the rate at which we delay, or how much we delay the signal here. It does sort of coincide with what we need to have happen with a higher or lower heart rate. So we alter heart rate at the SA node, right, by changing the action potentials there. This one lone axon coming down here, right, this guy coming down here and innervating the ventricular cells directly is not going to alter rate. It is going to alter contractility. Right? So the sympathetic innervates the ventricle and can alter contractility, and therefore how hard it contracts and how much blood it moves with each contraction, whereas both systems, both divisions of the autonomic nervous system affect the rate through their innervation at the SA node. Right? And in a separate video, we'll talk about the chronotropic and inotropic effects. Chronotropic meaning rate effects up here at the SA node, and inotropic effects down here, right, releasing again neurotransmitter. It would be norepinephrine because this is sympathetic. The parasympathetic would be releasing acetylcholine here instead of norepinephrine, right? Norepinephrine, sympathetic, acetylcholine, parasympathetic at the effector tissues. Okay, and again, more of that in the cardiodynamics video where we talk about control of how the heart beats, how hard the heart beats, inotropy, and how fast it beats, chronotropy.